of our graduate seminar series. The graduate seminar series is an event for the students of the graduate school of the chemical engineering school. And this event was created for you. This is a space for you to see a seminar from an invited speaker that has some experience working abroad. And then you can train on English, you can make questions, you can hear about his research, and so on. And of course, this is also for networking among yourselves. Okay, it's not, please avoid staying only within your lab. Try to meet people from other labs, and this is a space for that. So after the event, we have a coffee break outside, being served outside, so you can enjoy some pão de queijo and carrot cake, if I'm not mistaken, and orange cake, okay? And coffee, of course. So I think that's it for introductions. Let me go for the invited speaker. So our distinguished guest today is Dr. Thiago Ouriques Machado. He holds a PhD in chemical engineering from the Federal University of Santa Catarina. He has research experience at the Max Planck Institute from Germany. He was a former research fellow at the University of Birmingham in the United Kingdom. He was a Marie Curie fellow and former researcher at the RCSI in Ireland. And currently, he is an invited researcher here at the School of Chemical Engineering. The title of his talk today is Sustainable Polymers Design, Biomedical Agriculture, and 3D Printing. Topics include sustainable polymers from renewable sources, polymer networks and recyclability challenges, dynamic covalent bonds and circular materials, closed-loop recyclable photopolymer resins, and applications in biomedical agriculture and 3D printing. Thiago, on behalf of the director of the School of Chemical Engineering, Professor Dirceu Noriller, and the dean of the graduate program, Professor Melissa Vieira, I'd like to thank you for being here today. The floor is yours.
So, good afternoon, everybody. Thank you for the nice introduction. So, yeah, I've just started as a guest researcher here at the School of Chemical Engineering at UNICAMP. And this is an introductory talk about my research that I've been doing for the past 10 years, let's say. So, as Professor said, it's called Sustainable Polymers Design, Biomedical, Agricultural, and 3D Printing Applications. So I started my academic career at the Federal University of Santa Catarina, UFSC, where I got my PhD and my master's. In my PhD, I had a secondment to the Max Planck Institute for Polymer Research in Germany. And then I did a postdoc at UFSC during COVID. Then I moved to the UK as a postdoc, and I got a Marie Curie Fellow over there, so I stayed in the UK for 3 years. And then I moved to Ireland to work in the Polymers project. And now, well, I'm here. So, yeah, some data on plastics. I'm going to start with that. So, we know now that plastics has this huge environmental problem. And so just let's talk about some numbers. So when plastics started being produced in the 50s, they were about 2 million tons produced per year, and now we have 200-fold increase in that amount being 30% single-use plastic. So, for example, food, packaging, and so on. And only 5% is bio-based, which means non-fossil derived. And out of that, only 9% is recycled, and the rest is landfilled, incinerated, or is just leaked into the environment. So as a matter of comparison, for the greenhouse emissions, the entire life cycle of the plastic industry, from cradle to grave, generates 1.8 gigatons of CO2 equivalent. That's 1/3 of the road transport industry that basically burns fuel. So, as estimated for the 2060s, without any new policies, only 14% of the plastic will have a circular life. So that's a little bit worrying. So we knew, well, in the past, plastics were seen as this marvel material that we could just use and throw away, and they were lightweight, they were have good performance, and now we know that they cause ecological impact, and there is some waste management challenge. So this is called the linear plastics economy. And what I try to develop is the circular plastics economy, me and other researchers, of course. So sustainably sourced monomers, sustainable polymer synthesis and manufacturing, and the design of these plastics to be degradable, but benign environmental degradation, and also to be recyclable by design. So the motivation for my research is basically advanced circularity in polymer technologies and foster sustainable development. So I started the synthesis or working with this renewable polymers using vegetable oil and basically sugar, but in general lignocellulosic biomass. So vegetable oils are made out of triacylglycerides, and lignocellulosic biomass has sugar like cellulose and lignin in their composition, and they are vastly available, they're inexpensive, they have inherent biodegradability, low toxicity, and very interesting stereochemical that we can use for polymer chemistry. But how do we get polymers out of that? So just a second. Sorry. Through chemical modification. So we have the bio-based raw material, or actually the natural raw material, and through some chemical modifications, we are able to make sustainable monomers. So for example, triacylglycerides, they have many reactive sites, for example, ester, alpha carbon, double bond hydroxyl, omega carbon, allyl carbon, and all of them can go through chemical modification to make some monomers. For example, polyols, methacrylates, epoxides, they can go under polymerization and make new materials. And sugars, they are monomers by themselves because even the naturally occurring sugars, they can be polymerized into polymers. However, sugars are multifunctional molecules, and because of that, sometimes it's a bit hard to work with them because you can have side reactions or you can have undesired cross-linking. So it's very common for the sugars have their functionality decreased. For example, using bifunctional sugars such as the dianhydroalditols. And then you can create also a variety of monomers by reacting these two hydroxyls here. So the building blocks that we used in our work was 10-andesanoic acid derived from castor oil. In Portuguese, we call óleo de mamona. That's used for biodiesel here. And it's non-food vegetable oil. Propanediol, that's derived from glycerol. Isosorbide, that's derived from cellulose and starch, preferably from cellulose because it's waste, right? From like sugarcane bagasse and stuff like that. So the monomer synthesis steps very easy. It's just the Fischer esterification of the alcohol with the carboxylic acids using PTSA as catalyst for isosorbide and propanediol. So here we have the diene monomers, or alpha omega diene monomers, because we have the double bonds at the end of a very long fat acid chain. And then the polymerization step is the thiol-ene polymerization. So it's co-polymerized with dithiol. And we did this in mini emulsion because we wanted to generate some polymer nanoparticles. So we generate the macro emulsion with stirring, then add the dithiol, input shear energy in the system, have the formation of the mini emulsion, mini nanodroplets, and then thiol-ene polymerization occurs inside the nanodroplets, generating polymer nanoparticles. So here we have the TEM of the polymer nanoparticles, some of the samples that we've made, and we could see that there is some nanoconfinement effect in which the molecular weight of the polymers from mini emulsion are higher than the polymers generated in bulk polymerization. Can you hear me well? Because sometimes I move the microphone around. later on, we did some degradation studies with accelerated hydrolysis using acidic medium and also lipase, an enzyme. And after 12 weeks, in the acidic medium, the polymers degraded, they decreased 40% of their molecular weight, and in enzyme was much, much faster. So in 3 hours, the molecular weight decreased 70%, in 10 days, 90%, which is quite good, actually quite interesting. And the control was just PBS. That didn't change much. So later, we started the biological characterization. So we encapsulated coumarin-6, that's a fluorescent dye, to study the cell uptake with confocal microscopy. And here the cells are blue. The lights not helping, but the cells are blue and the particles are green. And we can see that the particles are either outside the cells or inside them. So we can see some cell uptake by the cell lineage, which is L929 or murine cells. And then we also studied the hemolysis to see if the blood was stable when incubated with the nanoparticles. And even at high concentrations of 200 micrograms per ml, we could see that the blood cells were not broken, because here the plasma is clean. And if it was hemolytical, then the cells would break down and the plasma would be red. And also the viability of these nanoparticles were high, so above 90% for 50 milligrams per ml. And these are the morphologies of the cells before and after the incubation. And finally, we use those nanoparticles to synthesize hybrid nanoparticles for hyperthermia. So we encapsulated superparamagnetic magnetite nanoparticles. And here we can see some TEM pictures where the black dots inside the big polymer ball is just the superparamagnetic magnetite. And even after encapsulation, the nanoparticles presented superparamagnetic behavior, and they were used for hyperthermia treatment, in which we applied a magnetic field to the cells with the nanoparticles that was applied intermittently, and then there would be a localized heat, and these heat would kill the cancer cells. In this case, it was HeLa cells. So then I moved to working with nanocarriers in agriculture. And why is that? So only 10% of the pesticides that we use in agriculture hits their target. And that's a very small amount from 4 million tons of pesticides we use every year. And this causes us to use more pesticides than we need, so pesticide overuse. And of course, because 90% of that misses the target, we get soil and water contamination, bioaccumulation in the food chain, pesticide resistance, ecological disturbance, and of course, ultimately, it's a threat to human health. So we try to solve this problem by using biopolymer nanocarriers. And there is a pool of biomaterials that we could use for that. And once we have the agrochemical inside the biopolymer shell, we have the sustained release of agrochemicals. And also the nanocarriers, they allow for efficient uptake, a specific targeting, and because they are made out of biopolymers, they are potentially biodegradable, and after degradation, they can reintegrate the geochemical cycles in the Earth system. So as the diseases we studied for that, they were caused by trunk-eating pathogens, so they eat cellulose and lignin, and they don't respond well to spraying because they're inside the wood. So the spray cannot reach there. And they're a big problem, especially in the south, for the grapes and for the apples. So we have Petri and Eska disease that affects grapevines, and European canker that mostly affects apple trees, and they are caused by fungi. So they affect also the fruit production, cause tree death, and also the rotting of the fruits, which is a shame because it's food. So the concept of this strategy was to use the same substrate, so in this case, lignin and cellulose, to compose the nanocarriers. So when the fungi releases the enzymes, instead of degrading the substrate, it degrades the nanocarriers that are loaded with fungicides, and then the fungicides are released, in loci, and the fungi dies. And we even did some studies in the field injecting latexes or nanoparticle dispersions in the trees. So how do we make those particles? So we chemically modified lignin with metacrylation. So we have Croft lignin here that's derived, that's actually a waste from the paper industry. And then we do a metacrylation reaction. And after the metacrylation reaction, we got 90% of metacrylation calculated by NMR. And to make those particles, we use mini emulsion polymerization as well. And so we have the lignin metacrylate in the organic phase, and also the encapsulated fungicides in the organic phase. And then we use amine crosslinkers with different functionalities to do a Michael addition cross-linking and form the nanocarriers. So, and for cellulose, metacrylation of cellulose was a little bit complicated, so we used a fatty acid instead, so andesonic acid that we've used before in the previous work. And we did a esterification, so we got 78% of degree of substitution, so 2.3 out of the 3 hydroxyls per unit was esterified. Also, we calculated it by NMR. And the cellulose nanocarriers was synthesized using mini emulsion polymerization as well, and this similar system where the encapsulated fungicides were added to the organic phase. And here we use thiol polymerization because this double bond is not reactive for Michael addition. So we use free radical thiol. So these are the particles, lignin nanoparticles and cellulose nanocarriers here because they're loaded. These are TEM images. These are SEM images. I would say the diameter is around 200 nanometers. And the encapsulation efficiencies were above 70% for all formulations tested. So high encapsulation efficiency, I would say. After we got the carriers, we tested their activity. For lignin nanocarriers, we targeted Petri disease, and we did an assay with the spores of the fungi. I call it germination and mycelial growth assay. So the fungicides or the nanocarriers were always tested against the free fungicide, and the nanoformulations were up to 80% efficient. So they inhibited up to 80% of the germination and growth. Here, the free fungicides have a better activity because they are free. They don't need to be released in the medium. So we thought maybe we should do a different study because let's see if it works in vivo as well. So we did a study in vivo for 4 years, more or less, and we did a score for the symptoms of the disease. 0 means no symptoms, 5 is a dead plant, and in the beginning we have the control that's not treated and the plants treated with the nanocarriers, and they are only treated once, so the nanocarriers are injected in the trunk only once, and after 4 years the control got sicker and the plants treated with the nanocarriers were healed, fully healed, no symptoms at all. And for the cellulose nanocarriers, we did a similar study in vitro only with the encapsulated fungicide or the nanoformulations being up to 70% inhibition. And we tested against non-cellulolytic fungus. So this fungus here is not, it cannot degrade cellulose because it doesn't produce cellulase, so the enzyme that breaks down cellulose. And the inhibition that we see here from the nanocarriers is just because of the diffusion of the fungicide. So the inhibition here was much lower, so only 30%. That's because of the diffusion. So it means here that if we exclude the diffusion, we still get further inhibition, and that's because of the degradation of the nanoparticles by the fungi. So moving on, when I moved to the UK, I started working with additive manufacturing, also known as 3D printing. And 3D printing is basically when we have a CAD model on the computer, and then we can translate that into an object using a 3D printer that builds the object layer by layer. And it's an assembly-free type of fabrication. It uses less material and minimal waste compared to the subtractive manufacturing, that's the regular manufacturing system. And also it balances supply and demand, which means we can manufacture on demand, which is also more sustainable. So the market size projection of 3D printing goods for the 2030s is over 50 billion USD. So it's a growing market, and we can see some products already in the market, such as aerospace parts and automotive parts, consumer goods like these Adidas shoes, or also dental and biomedical products like dentures. But the type of 3D printing that I study is light-assisted 3D printing. So we cross-link polymers with light. It's a rapid processing. We can make complex geometries. It's we have higher precision, and we can also make very tough constructs. So there's many types of light-assisted 3D printing, but I work mostly with VAT photopolymerization, DLP. There's also stereolithography, but I work mostly with DLP, in which we have the resin inside a vat, and then there is the building plate, and the resin is between the building plate and the source of light. The light is irradiated into this gap here, and then we cure the construct layer by layer. But what's the problem with that then? So the photoresins that we have that are available in, commercially, they are mostly based on acrylate chemistry. So which means that once the photoresin is cured, we have this network here that it cannot be recycled. So it just goes to landfills. So not very sustainable. And also the monomers and the diluents, they are also petroleum derived. So we have a problem of source and a problem with end of life. So we need to fix that, but how do we do that? So what researchers have been doing is trying to make resins based on, bio-based compounds. So for example, itaconic and succinic acids, or terpenoids, vegetable oils, and lignin. We've done some work in that sense as well. So we made some monomers from itaconic anhydride and perolyl alcohol, which is a terpene, by esterification with an enzyme. And then we made a resin with a thiol crosslinker, BAPO as photoinitiator. And here, because we have this acrylate-like double bond, we have step growth from thiol-in polymerization and chain growth from the acrylate from the itaconic acid. So it's a mixed mixed mechanism system. So we were able to cure the liquid resin into a photocured film. However, this formulation here has a very high viscosity, misspell here, viscosity too high for DLP. So we had to include some reactive diluents to make it less viscous. So we used other terpenes, so parallel alcohol, limonene, and linalool. Something happened here, it's erased, but still, that's limonene. So we compared the mechanical properties varying according to the diluents. And as we can see here, we can see that the resin with limonene as diluent had the best mechanical properties. So that's what we chose to print. And so we ran photorheology to help to figure out the parameters for 3D printing. And then we are able to 3D print some constructs. And this is the photo of the printing, and this is the 3D microscopy images that we took, optical microscopy. But this is still not recyclable because those bonds, they're not going to break so easily. So acrylic chemistry or olefin chemistry, whatever, the enthalpy overweighs the entropic penalty. So they're pretty much non-reversible. How do we make them reversible? So actually, we need to use dynamic bonds or dynamic covalent bonds in which the enthalpy and entropy are closer to an equilibrium, and you can just tip the reaction into the forward or the reverse direction. And there are many examples of that in the literature, but mostly the dynamic bonds are inserted as preformed bonds in the monomer, and the network forming functionalities are still acrylates and epoxides and olefins. So even though they're cured and they can be depolymerized because these are dynamic, in order to recycle the material, you need to add more monomers because you don't have the network forming functionalities anymore. And so we have examples for hydroxy esters, thiourethanes, thioesters, and this just creates a snowball effect in which, in order to recycle the material, the only way is creating more material, and in the end, you just postpone the problem. So what we did was to work with disulfides. So we have this bio-based molecule, it's lipoic acid with a cyclic disulfide, and it can polymerize into a linear disulfide. And this is completely reversible, and it's biosourced, and it's inexpensive as well. You can buy it as a supplement. So previous work in the Dove group designed these networks that were completely depolymerizable, and they could go back from the film into the resin just by diluting it down. Later on, the Craig Hawker group, they started making these bottle brush with lipoic acid attached to PEG and cross-linking it with PEG diacrylate. But they were not able to fully depolymerize it, and which wasn't expected. So they couldn't recycle it. They just were able to break it down to oligomers. And we knew for a fact that we could fully depolymerize those materials, and we thought, why? So we did a calculation, and that's because we had more sulfur in our system than they had. But inspired by these works, we decided to design a fully polydisulfide network based on lipoic acid. So we just made some monomers with biosourced alcohols. Simple procedure, just changing the alcohol to the esterification here. And then we can create a fully polydisulfide network through radical ROP of the diethylene ring. So first we made some monomers and investigated printing parameters. So here we have the main monomer, which is the glycerol from glycerol trilipoate and ethyl from ethanol lipoate as a linear monomer with 38%, and the triarm 78%. And we printed these arrays of square bridges and walls to test the resolution. And we did that by printing the material and then taking some pictures with the optical microscope, measuring the squares and the bridges and everything, and then comparing to the model and see how the measurements deviated from the model. So we got very good precision. And then we started printing more complex structures like the Benchy boat, Eiffel Tower, some scaffolds, the Old Joe from the University of Birmingham. Later on, we tried modulating the properties, changing the substituents here. So when we have some cyclic linear monomers, we can see the increase in the glass transition temperature. The same happens for the cyclic crosslinker. When we have the long alkyl chain in the linear monomer, we have some crystallinity in the material. And of course, that also affects the mechanical properties. So stronger materials with cyclic moieties, weaker materials with linear moieties. So we can get a full range of different materials by changing the alcohols. So after that, we started doing the depolymerization and recycling. So basically, we got the prints, we grind it, and then we depolymerize it by just heating them up in DMF. So we have a ring closure reaction here, and the network goes back to the initial monomers with 90% yield. And then we are able to use the resin again for reprinting. And here we can see by NMR that the resins in the pristine first recycle and second recycle are basically the same. So as far as composition goes, they're very similar. So comparing the content of crosslinker, we have 70% for the pristine, 68 and 69 for the recycles, and the cyclic disulfide is recovered by 96% and 94% respectively. So we have 96% of this in the first recycle and 94% of this in the second recycle, which means that we still have some oligomers that we can see by the GPC traces. So even though we have the 4 or 5% of oligomers, we are able to recure it, and we get thermomechanical properties very similar to pristine. Then we did the same precision study with the recycled resins. We also get very good precision, and we were able to also print some more complex structures. So pristine first recycle and second recycle over there. And this was pretty much the first work to be able to fully recycle 3D printing resin for DLP. So after the Marie Curie Fellowship, I moved to Ireland to work in the Polymers project, which is, well, I'm going to talk about it in a few moments. So basically the project is to make some commercial biomedical materials from 3D printing using amino acids or polypeptides. So I was working with this first area here, developing materials from amino acids and trying to work out 3D printing resin that later would become tracheal implants. So it was a big project with 8 organizations across 6 countries. And so to make those materials, polypeptides, well, I think, so it's obviously they're bio-based and they're body-like macromolecules, even though they are synthetic, and they are suitable for biomedical applications. It's well documented in the literature. But how do we make those materials? So we got the amino acid, we make N-carboxy anhydride with trifosgene, and then we do an ROP with base or nucleophile. But can it be light triggered or triggered by light? The answer is yes, but the first two works to do that, they used amine photo cages, but the quantum yield of the photo cage was not that high, which means that the release of the base upon photon absorption was not that good, and also the reaction kinetics was a bit slow or moderate because they use the normal amine mechanism because this is a initiator nucleophilic base. So how do we go around that? So we started in collaboration with our collaborators from Spain using photo-based generators, which has a higher quantum yield and is able to release a base in the system when it's irradiated with light, and it's amenable to visible light as well with the addition of a sensitizer. And this free base here is a non-nucleophilic base. So instead of the amine normal mechanism, we have activated monomer mechanism with a much more rapid reaction kinetics. So we started the study using BLG NCA because it's simpler, it's easier to make, and it's also stable. And we tried different photo bases with different conjugated bases here, different pKa's. And DBU, because it has the highest pKa, was the fastest as well. And as far as molecular weight goes, it didn't change that much, but DBU was not the highest score here. It was DMAP, but DMAP's much slower, and we decided to keep the kinetics over the molecular weight, because if you're going to use for printing, you need very fast curing. So we studied the dependence of the kinetics with the monomer concentration, higher concentration, faster kinetics, and also higher concentration of monomer, higher molecular weight. Light source dependence, even though it could use blue light, 4 or 5 nanometers, the UV was more effective, and also the UV provided higher molecular weight. The blue light provided higher molecular weight, but as we said, we were actually preferring faster kinetics over molecular weight because in the end we would have a network, so we wouldn't make that much difference for this first stage at least. So to make the networks, we designed this cysteine-derived bis NCA, which has a disulfide bond here, my old friend disulfide bond, and so we were able to cure these two monomers to create the network, and we compared only the the bis NCA with the addition of BLG NCA with 4:1, so 4 bis NCA, 1 mono NCA. With the addition of the mono NCA, the reaction is faster, but again, we are interested in faster kinetics, so this is actually a nice result. For photorheology, it didn't change that much the gelation time, but we could see that the bis NCA by itself had a better mechanical properties. So, and finally, we prepared some networks for degradation studies, and here we only used the bis NCA because we wanted a plethora of disulfide bonds, so we made those films. Oh, the scale bar here is 1 centimeter. It's missing. And then we did a disulfide reduction to change the disulfide into thiol using the TCP reagent. So what was insoluble and DMF, it's swollen and DMF here becomes soluble. And we can see that, well, we cannot measure the diffusion coefficient for the network because it's not soluble, but we did for the monomer and for the recovered oligomer. And the monomer has a diffusion coefficient around 10 to the minus 6, and the recovered oligomer 10 to the minus 7. So it has a lower diffusion coefficient, which means it's slower, therefore a bigger molecule. And then we can see that we have actually an oligomer instead of a monomer, or, well, the network would be insoluble. And we can also match the protons in the NMR. So as a summary of this talk, I've presented some polymers and resins, 100% from renewable feedstocks, used to make films, resins, constructs, and nanocarriers. The resins were 3D printed in high-resolution parts. I showed also a broad polymer chemistry bio-based platform with varied properties for several applications, and also showed the degradation of bio-based polymers and the closed-loop recyclability of networks. What's next then? So I'm here at the School of Chemical Engineering at UNICAMP. I returned to Brazil with a fellowship from the CNPq. They're trying to bring back some researchers that were abroad. And here, I'm very grateful actually for the school being like hosting my research here, and also Professor Ouriques for hosting me in the lab. And here I'll be studying sustainable and bio-based materials for several applications as well, biological 3D printing, recycling mostly. I'm really into this theme at the moment. And structure property relationships of polymers like stereochemistry, how that affects the properties. So I'd like to thank the institutions I've worked in, all the collaborators for that amount of work, and also the funding agencies. So thank you very much to you as well for your kind attention, and I'm happy to take any questions.
Absolutely. So questions? Who's going to start? Please, one at a time. Okay.
Okay, thank you, Thiago, for a nice presentation. I'd like to hear your opinion about what kind of building blocks do you consider most promising for developing this covalent, dynamic covalent networks that you mentioned, considering the Brazilian scenario, the Brazilian context? What do you consider the most promising that could be synthesized and derived these building blocks?
So I really like the idea of working with waste. So we produce a lot of waste from agriculture. So that's basically lignin and cellulose. So we could use those. And we also produce a lot of vegetable oil for biodiesel. So there is a lot of glycerol there. So 20% of the reject and biodiesel production is glycerol that could also be used. So, which is a boring answer because it's basically what I just presented here. But also terpenes, I think it's very good because it's also readily available. Let's see, maybe something that could be produced by fermentation using those lignocellulosic waste as substrate. But bioengineering is not my strongest suit. Let's see, I think that's it.
Next question.
Thank you very much. Very, very interesting. I have 4 questions, in fact. The first one is about the hyperthermia. What's the temperature that...
40 degrees.
40?
Yeah.
Okay. We were curious. We were discussing that
It was
just about 2 hours ago in a class.
I think it's 42 to be more
Oh,
exact.
okay, perfect. So the second question, do you think, I am very interested in these nanocarriers in agriculture. In Brazil, we are having a big problem recently with the vassoura de bruxa. Have you ever seen anything in this sense? Because this
I know.
vassoura de bruxa kills a lot of trees.
So I don't know about this one, but I know that there is a company in Florianopolis, the city where I'm from. What's it called? I forgot. But they are working with some nanocarriers for agriculture over there. I don't know what they're targeting, but I know that they are working with it. Well, I can look that later because I don't remember the name of the company. But I don't know I don't think there is anything commercial so far. But
because this would be quite interesting because this attacks chocolate. I cannot imagine the world without chocolate. And they said that it could extinguish
yes.
chocolate.
Oh, I see.
So it's something that we would be very interested in.
Yes.
And have you incorporated any kind of dye or ink in your inks for 3D printing?
So what we did, it's because sometimes we use dyes, but to restrain the light. Because sometimes we have the curing and the, let's see if we can. Oh yeah, we can show it here. I can find a very good picture. Yeah, so here, so this is the the print starts from here, right? So it's vertical, and we use a dye when the light goes too further, and it prints like big depth. So we need a dye in order to avoid the light penetration going too deep.
Okay.
And then we don't get like a very good precision. So in this case, there is no dye, but we used a dye in the terpene work. So it was a reddish dye, Solden red.
Okay, because I think that it would increase the commercial value of the the inks if you could incorporate stably the dyes in them. Oh, perfect. And last question is about hydrogels. Do you think that any of these formulations you developed, do you have one that would be more interesting for hydrogel application?
Polypeptides for sure. They are very used for hydrogels. Yeah, and they're very good for biomedical applications as well.
Okay, thank you so much.
You're welcome.
Thank you, Angela. Any more questions? Come on, guys. Do you have people from the undergraduate course here? Galera da graduação? Okay, some students. Guys, if you're interested in his work, you can ask him, talk to him, to see if he has opportunities for undergraduate research. And the guys from the graduate course, of course, you can talk to him to exchange some ideas, of course, based on his experience abroad. So this is very important for you guys that are working in the graduate course. Any more questions? No? Okay, so I think that's it. Another round of applause for our invited speaker. Thank you very much, Thiago. And we have a coffee break outside for you guys. Good afternoon. Just a second, let's take a picture. Ok, pessoal. Muito obrigado, pessoal.